Hello everyone. I'm very happy to have opportunity to represent our poster, Unravelling the Complexity of Cellular Interactions using Underlying Graph Representation of Single-Cell Transcriptomics Data. I'm Akram Vasighizaker, a PhD student of Computer Science at the University of Windsor, and I performed this project with Trina Horo and Dr. Luis Redo, my supervisor and the professor of the Computer Science at the University of Windsor. Let's begin with a brief introduction. The objective of this research is to discover and analyze intercellular signaling networks using single-cell RNA sequencing data and graph convolutional neural network. The proposed methodology, or we call it 6CCO, is to construct a cell-cell communication network from gene expression by embedding it into a graph representation. So in the graph, nodes represent cells and edge represents cell-cell interactions. Building cell-cell network using the similarity matrix based on non-cell-cell interactions, and then reducing the complexity of constructed graph attributes while preserving the global structure of the input graph using information gain dimensionality reduction in the pooling layer. So by extracting low-color subgraphs around target nodes, we can capture locality, capturing both latent and explicit characteristics of the graph constructed from the gene expression profile of individual cells, enabling the capturing of genes and network structural information from the data. Finally, deep graph convolutional neural network architecture used to predict potential interactions. In another word, formal problem statement, the problem is with the prediction of cell-cell interaction. Given a sparse, high-dimensional single-cell data, the output would be a likelihood to predict a likelihood of a link between two cells. So our solution is divided into two main steps. First, convert single-cell data to an undirected attributed graph, and then predict using a graph neural network. Let's move on to the methodology. This methodology includes two main steps. The first step is data preprocessing, which includes normalization, filtering, transformation, and quality control. The second step is to construct cell-cell communication network from single-cell data. And the last step and the main step of this proposed framework is to apply graph convolutional neural network on the constructed attributed graph from single-cell data and prediction. In more detail, an constructed attributed graph from the single-cell data includes three main information: node attributes, structural information, and node embedding. The graph convolution can be divided into four main steps. A linear feature transformation is applied to nodes information matrix X by multiplying it by a weight matrix W. And the node information is propagated to neighboring nodes as well as the node itself by multiplying it by its adjacency matrix. And then each row is normalized by multiplying it by an inverse of a degree matrix. A nonlinear activation function is applied to obtain output. So now I'm excited to represent our results. To evaluate the results, we use 6 public annotated single-cell datasets of human and mouse pancreas datasets. These datasets are extracted from NCBI Gene Expression Omnibus. And these are the statistical information from the network constructed from single-cell data. You can see graphs include different number of nodes and edges in different datasets. We evaluated the methods against other methods such as latent feature-based methods and the latest GNN-based approaches for link prediction in terms of AUC, ROC, accuracy, precision, recall, and F-score. Our proposed methods exhibited a performance of approximately 99% for all performance measures across all datasets, surpassing other methods. Additionally, our approach demonstrated the lowest false positive rate of 0.0135 among all approaches, indicating a very low likelihood of predicting non-interacting cells as interacting cells. And this is the result of comparison of the proposed method with GNN-based method based on AUC. And this is the result of ROC curves for two datasets of Barden-Human 1 and Barden-Human 2. Results of false positive rates against true positive rates for the same datasets, indicating a low rate of false positive rate for both datasets. The results of other datasets shown the same. Additionally, we run gene manual algorithm on the input list of top 20 genes that ranked based on a Wilkinson methods and found interesting results. Genes which are extracted from the pooling layer, indicating the most effective genes in the predicted information in the predicted interaction between cells. So as an example, working on Barden-Human 1 dataset represented key regulators and effectors of the functional relationship between cells in three subnetworks according to GO annotation. The first subnetwork is insulin-like growth factor binding. The second one is cellular response to type 1 interferon, and the last one is growth factor binding. All of these subnetworks showing those genes that have a key roles in cellular response to immune responsibilities. I would like to take this time to acknowledge our supporter and thank you very much for listening and have a great day.